Biochemistry Module Lecture 5, Lipid Structure and Function. We'll start off by defining what a lipid is. Lipids are defined as a molecule that is insoluble in water and soluble in nonpolar organic solvents. That's obviously a pretty broad definition, but when it comes to biological systems, there are three in particular that we will discuss. They are listed below: triglycerides, phospholipids, and steroids. And we'll talk about all three. Starting first with structural lipids. The primary structural lipid is the phospholipid group. These are amphipathic molecules that form lipid bilayers of cell membrane. If you remember, amphipathic molecules are molecules that exhibit the traits and characteristics of both hydrophobic and hydrophilic regions. Because of this, you can create very interesting little regions and pockets of distinction. These phospholipids are comprised of hydrophobic tails and hydrophilic heads, and the saturation of these tails is what determines the fluidity. Obviously, things such as cholesterol and other sorts of nonpolar compounds are going to affect the fluidity and the saturation. Glycerophospholipids are phospholipids with a glycerol backbone, and this is the one that you'll commonly see in certain parts of the body, especially things such as adipose. If we were to look at what these amphipathic molecules look like, we have a diagram at the bottom listed here of a basic phospholipid. But when you see cell membranes, they look something like this. You have the hydrophilic areas pointing out with the hydrophobic tails creating sort of a bilayer. We have a separate lecture talking about biological membranes, but it's important to mention what the primary component is in this lecture. Sphingolipids are basically phospholipids that contain a sphingolipid bilayer. They are often also phospholipids by categorization. Sphingomyelins are the major components of the myelin sheath, as the name would suggest, and the glycosphingolipids are attached to sugar moieties instead of phosphate groups. Gangliosides contain oligosaccharides with at least one terminal sialic acid. Gangliosides are especially important when we look at nervous system function. At the bottom right are some examples of sphingolipids. Waxes are comprised of long-chain fatty acids that are esterified to long-chain alcohols. They're used by organisms to protect against evaporation and parasites. This is largely because waxes are incredibly hydrophobic, meaning that they often don't let water through, which allows it to prevent moisture loss. At the bottom are some examples of waxes. Waxes are used in a lot of products, for example, things such as lip balm. Why do we use things like waxes for these kind of purposes? Well, obviously to retain moisture. Now that we've talked about structuring lipids, let's go into the next categorization of lipids, which are signaling lipids. Terpenes are steroid precursors, and they're made from isoprene. Terpenoids are derived from terpenes via oxygenation of the backbone and rearrangement. Terpenes in general have odoriferous traits, meaning they have specific smells. If you've worked with these in labs, you'll probably notice a lot of these scents. Terpenes are very commonly used in perfumes and colognes and other scented items for this reason. Steroids are one of the hormones of choice that are used by the endocrine system. Their oxidation state and functional groups will vary, but they have a very common characteristic. At the bottom are some examples of steroids. These hormones have high affinity receptors, meaning that they work at very low concentrations. This means that there does not have to be very much hormone to generate a response. And a lot of times, these steroids are going to affect things such as gene expression and metabolism, making them especially important. Cholesterols are, like I said earlier, important for membrane fluidity and stability, and they're often precursors to things such as steroids, bile, and other sorts of molecules. When we talk about lipids, steroids are incredibly important, and they come up multiple times, so make sure you have a good idea of what these structures are and what the composition and characteristics of these compounds will be. Prostaglandins are autocrine and paracrine hormones that regulate CAM levels. They can affect everything from muscle contraction to body temperature to circadian rhythms, along with pain. The fat-soluble vitamins are often characterized by letter, but they have more formal names as well. Vitamin A is also known as carotene. It is metabolized to retinal for vision. Cholecalciferol, or vitamin D, is metabolized to calcitriol in the kidneys. This is going to regulate calcium and phosphorus homeostasis in the given intestines, and obviously, because of this reason, they promote bone formation. Tocopherols, or vitamin E, is a biological antioxidant. The aromatic ring destroys free radicals and prevents oxidative damage. And lastly, vitamin K, or phylloquinone, and menaquinone are important for the formation of prothrombin, which will be important in clotting factors. The last categorization of lipids that we need to talk about are the ones involved in energy storage. And there's one in particular that we have to talk about, and that being triacylglycerols, or triglycerides. This is the preferred method of biological systems to store long-term energy supplies. It contains one glycerol attached to three fatty acids by an ester bond, and the structure of a triglyceride is listed below. Because the carbon atom in lipids are more reduced than carbohydrates, they yield more energy during oxidation processes. However, take note that these are incredibly hydrophobic as well. Adipocytes are simply animal cells specifically designed to store large triacylglycerols. If you hear the colloquial term fat, this is often what they're talking about. Although we often don't like to think about fat and want to look as fit as possible, some amount of fat is very healthy and important for long-term usage in energy supply. The final topic we'll go into is saponification. In general chemistry, we also touched base on this, but let's go back to it. Saponification is the ester hydrolysis of triacylglycerols using a strong base, the reaction being listed below. Lye is obviously the most commonly used one. And micelles are shells of fatty acid core that are formed when using a soap. This is how we can dissolve hydrophobic compounds in water. So if I were to look and try to wash something, soap molecules will go around something that's hydrophobic, like so. Very similar to a membrane, the hydrophobic layers are going to develop an affinity for this given compound and encapsulate it in a spherical capsule. This allows it to be wiped off and carried away in water. We'll finish our discussion of lipid structure and function there. In the next lecture, we're going to go into some interesting developments involving the genetic code and biotechnology.